Recording. Alright, so just a short recap of previous lecture. We've discussed different mass spectrometers. We discussed how they work, but for us as biologists, it is not the technical details which actually matters, but what we can do with different types of mass spectrometers. We've already seen that the most used one in proteomics is an orbit trap because it has high resolution, high sensitivity, can work either fast but not so accurate, or slow but really accurate with high resolution, with high sensitivity, and so on. We've also discussed that we have two different ways of ionizing our proteins or peptides of interest. It's electrospray ionization for chromatography equipment, because we can just get chromatography equipment here and just put all that we have, all that eludes from the column, just in our mass spectrometry equipment right away. And we have so-called mild ionization, where we can put just drops of our samples, dry them, co-crystallize them with some matrix, and this mainly used with identification of spots from your gel, mostly. Also, we've discussed that there are different problems with resolution, with interpretation of spectra, and we've ended up with the mean of being able to identify our peptide just by so-called triptych peptide fingerprint. We have just a triptych small peptides. We can search the database, either some homemade database from just a FASTA file, or some commonly accessible database as a SwissProt and so on. Today, we have to discuss additional approaches which will allow us to not just annotate our proteins, but identify them, be able to actually see primary sequence of our peptides. So, just to start, we have to start from explaining so-called tandem mass spectrometry. We've already touched this topic yesterday when we were discussing applications of so-called quadruple mass spectrometry. We can filter some ions, and we've also touched this topic slightly when we've been discussing so-called ion trap mass spectrometers, where we can isolate just one type of ion, then fragment it, then see what was the fragment, or even get rid of all the fragment except the one, and this fragment we can again fragment in a smaller fragments. So, why this tandem mass spectrometry and this fragmentation of big importance for proteomics? That's because actually this fragmentation doesn't happen in random fashion. For instance, if you have a set of bricks for some Lego toy, you can make different types of machines or whatever from the same set of building blocks, right? And let's say you have 100 machines that you made which are all the same, and 100 of machines which are a little bit different, and you will just start throwing from 1 meter on the floor. They will fragment in some way, right? But will this fragmentation be, let's say, random, or will there be any rules of this fragmentation? For instance, one machine will have some long stretch of the same blocks. Will there be, will it provide more case of fragmentation of this long structure? And another machine will be more or less spherical, for instance, and it will fragment harder, but the fragments will be different, right? You've used the same building blocks, the same number of them, but you've just connect them in different way. And after fragmentation, you actually, after having a lot of such fragments, you can decide what was the machine before you fragmented, right? The same is true for most biopolymers, because in biopolymers, it's the link between monomers which fragment more easily than those building blocks itself. For instance, for nucleic acids, it's those nucleic acids, and the bond between them is easiest to be fragmented. For proteins, it of course our peptide bond, which also can fragment easier than any other combinations. Just for some oversimplification, we can get a number of fragments. We know the mass of each amino acid, and we can actually, even for some really good peptides, try to derive the sequence that was on this peptide before we started fragmenting it. In most cases, it is impossible to do because not all of those fragments will have charge on them, and some of them will be missing. For instance, if you have a proline, there will be no good fragmentation of peptide bond between proline and other amino acid. Why is so? What's important about proline?
It is amino acid, not amino.
It's secondary amino also, so it has a structure which can then just regroup, and we will just actually lose this fragment from interpretation. It's so-called proline gap. So if you will ever encounter this abbreviation, proline gap in mass spectrometry, we now know what is it. So, why it is quite important also? Because we've got those tryptic peptides after digestions of our protein or proteins. We know that there are at least some amino acids at some end, because trypsin cuts at some predefined positions. But we don't know actually what is the sequence of those peptides. And if you can then select just one peptide of this mix and then fragment it, in ideal case, we will be able to get information about its sequence. Of course, in ideal setup, it's not always possible. But if you have, let's say, two unique peptides for some protein, can you say that you not only annotate it, but actually identified this protein? I think not probably, but we can actually say that the identification of this peptide was made by two, and then provide you peptides, which, and so on, so on, so on, so on. So actually, nowadays, two unique or signature, it's the same name actually, peptides is enough to say that you have identified the protein. Not protein form, because those unique peptides can be different parts of your protein, and probably some post-translation modification are between them. So, not protein forms, but proteins, yeah, just two unique peptides, or even one unique peptide and two non-unique peptides. Also, not quite good, but it's possible to publish such results. So, how it done actually? Mostly in so-called orbit trap, it can be just programmed to scan all the peaks here and then select those peaks and do the fragmentation for them. Nowadays, there is another technology, is so-called quadruple time of flight mass spectrometry, which have some additional benefits. It's not a mainstream yet, but probably it will be, and it's easier to understand how it's done. We have our ionization, we have first separation, for instance, mostly it's quadruple separation, where you can ionize or select only one peptide, or actually one M/Z ratio. Then you have collision cell, which is also in fact a quadruple, just filled with some gas, be it nitrogen or argon, or even something heavier, but mostly nitrogen, it's cheaper. And then you have other step of M/Z separation. Nowadays it mostly either orbit trap or time of flight. Time of flight actually have some benefits here, but not mainstream yet. And a detection for orbit trap mass spectrometer, separation and detection is in the same volume. In time of flight mass spectrometer, of course, we have a special detector. It wasn't clear why we need it and how we can do such an approach. Okay, then. Actually, what I've told just 5 minutes before is an oversimplification, because we have this peptide bond, but it can be fragmented at different positions. For instance, here is our amino acid here. Here's another amino acid with another radical, and we can have breakage of our bond at 3 different positions. And also, those 3 different positions provide us with 3 different ions, be it one ion from the left side or any other ion from the right side. So actually we have series of ions, and the nomenclature is following. If we are talking about this left side part, it would be A type of ion, if the bracket here, B, C. From this position it would be X, Y and Z. So just to name them. And actually, fortunately for us, in certain types of fragmentation and in certain type of mass spectrometers, there are some predominant ions which we will more likely to see than others. So actually it's a little bit easier because mostly we will have so-called Y and B series. Let's see here, Y and B. So this is actually the position where the breakage of peptide bond is will mostly occur. It doesn't mean that there wouldn't be any other series of ions present, but mostly in electrospray ionization and collision fragmentation we'll see this kind of ions. So let's see some really nice spectra. It's not always you can get something like this, but let's see. We have some peaks, and there are some difference in mass between those peaks. And if we'll just see which amino acids can fit in, concerning that we have only Y and B series of ion, we can actually, for this small peptide, get positions and amino acids here. So, as you can see, it is possible to somehow, in some cases, to do de novo sequencing on peptide, but not always. With proline, there is a gap. Leucine and isoleucine have the same mass exactly, so it's not possible to distinguish between them. And also, the pairs of some amino acids, though those amino acids are different, will have the same, almost the same weight. So there are problems. And also, we can see this example where we see not only Y but also B series of ions. Why having both series of ions is of advantage for us?
Maybe the second series differs.
It doesn't differ actually much, but it be possible that, for instance, Y series of ion is inhibited by some properties of our peptide, but we'll get those three series of ion, for instance. So actually, if we want to annotate, or actually we ask computer with some software to do so, it will try to use both series. And also, what do you think? Is it possible that some amino acid will be fragmented from both sides and will have just an amino acid? Why not? If you have excessive energy, and after first round of this breakage of bond, there is still enough energy to break some other bond, it is also possible to have those ions. Unfortunately, here on those graphs, it's not possible to see them because we have only 200 m over z, but it also can happen that it can allow us to identify either some certain amino acids in this sequence, right? Because we have those Y and B series, and also so-called ammonium ions, which are those amino acids which were broken from both peptide link was broken from both sides. So that's how it looks like, and also I've already covered this ammonium ion, and as you can see in a small m over z weights here, we can also have those ammonium ions, and if we have one, it is proof enough that at some position there is such an amino acid in some in sequence of our peptide. Of course, it can be nice for us, for instance for proline, for some other ammonium ions. And here you can see this formula, monoisotopic mass for different peptides, and of course we can get stable where we have ammonium ions and cleavage of radical groups, because for some amino acids even radical group can be cleaved in more parts, and it can allow you to be even more sure of your identification, but it's not always possible. What I want to discuss here is that we've discussed different types of mass spectrometers, and I've stressed that some of them have difference in being able to see those small m over z ratios. For instance, ion trap will lose some of them, orbit trap mostly will lose all of them. So those small masses will be mostly accessible at time of flight mass spectrometry, which can be one of the benefits of using them. So, just to make things more clear, if we have some sequence, be it this sequence, we'll have different series of ion, B and Y ions, and you can see that since we cleave it and have one part of B ion, one part of Y ion, we'll have two different weights here, and a weight of B ion, weight of Y ion, and also, as you can see, the intensities of those fragments, or the number of those fragments, count of those fragments if you will, is different. Why do you think not all ions are present at the same amount in this fragmentation spectrum?
Because some parts of amino acids are the same?
No.
No?
Some of them, as you can see, have almost zero intensity from the real data. From the data, if we'll just do it in silico, we'll get just the same level. What do you think about charge? Is there some preferential for charge to move during this fermentation phase? If you, for instance, have some basic amino acids and acidic amino acids, and you want to get those ions which have excessive protons.
Negative charge to
Right, absolutely. And we've also discussed proline, which can get just without charge, and if it is not charged, we can detect it, right? We'll have just some loss in mass between two peaks, but it could be proline and it could be something else. So, in real mass spectrometry data, we will have something like this, which sometimes will be enough for the de novo sequencing, but most of the time some part of your peptide will not be seen on this mass spectra, and you will have to do some other approaches. What can be done if you still want to get the sequence of your peptide? What do you think? For instance, if you have some protein, you have no means to do Edman degradation, which will be fine, because at least 20 amino acids will be sequenced at really high quality. And you have only your mass spectrometer, your protein, let it be a lot of your protein, a gram of your protein, so you can do everything you want with it. And you want to get as good representation of the primary sequence as you can. What will you do? Here we have only tryptic peptide. Can you use different protease, for instance? So we've already talked about protease profiling, right? And if you have some problems in sequence detection, you can just use different protease, for instance, to see whether there is this amino acid, because there is a lot of proteases, and some proteases can happen that they will cut after the amino acid that you have already identified, but before the one that is not yet identified, right? So you can use information of selectivity of those proteases. Also, actually, you can add some chemicals in your solution which will act as a donor even for electrospray mass spectrometry, as a donor of those protons, and you will actually, in fact, make those ions more. The count of them will be more. You will be able to detect them, so they will be more intensive. So there are ideas how to overcome this problem, but mostly you will get this kind of information. You can try to do so with those links. It's a link for peptide cutter with different proteases. This one is for making some mock mass spectrometry data in silico, so you just get your peptide. In this program, we'll use a lot of different approaches to predict not only fragments but also their intensity, for instance. And you can use this program to actually try to identify them. So, what we can have in our spectrum. This is quite nice spectrum for fibrin peptide B, which fibrina peptide, which actually used as a calibration standard for most of mass spectrometers, so we will use it a lot. And the problem here is if those ion series haven't have any color on them, would you be able to just by hand assign them? What do you think? For instance, here's one peak which is quite big, but is not assigned to any of those ions. There are some small peaks which are smaller than the neighbor peaks, but which is assigned and so on, right? So, if you did not know the sequence of your peptide before, it will be a bit troublesome to do de novo sequencing from this data. And what is the source of those ions, those M/Z ratios, which are not annotated here? There are a lot of probabilities. For instance, some chemicals after ionization will lose some parts. Usually it's water molecule which tries to just go away from your ion, and you have this difference of mass. Phosphor, so it really likes to leave your ion, and some other chemicals. So after ionization, it can be that the mass of your molecule will be a little bit different. Something will be lost during this process. Also, we know about adducts. Something can be added. For instance, we've already discussed sodium, potassium, but there are a lot of different adducts. For instance, you use just acetonitrile, and acetonitrile can somehow be ionized with your ion of interest, adding some different weight. Also, it could be contamination, it could be some other fragment. I've already said that B and Y series are the most frequently seen, but there are also other ions which can be seen from this series, like A, C, X, Z, ammonium ions, and so on. Also, it can be that, for instance, this peak can be fragmentation of the side group of your amino acid, and you have this difference in mass. And also noise, either chemical noise, something that is in your solution, or electronic noise, it's also possible. So, the questions we have to discuss here. Is it possible to de novo segment every peptide after this fragmentation?
Yes.
No.
There are two different opinions. Please elaborate. Why it is not possible or possible to do? I've mentioned that this is an ideal picture of something that is just used as a standard for calibration of mass spectrometry, which is all presented in a really nice solution without any problems, so you know actually beforehand its sequence, because it's some standard which we use for calibration.
We cannot, because if we don't know the previous sequence.
There is a chance that we can, but not every fragment and every peptide, right? So we can be lucky enough, and it can be possible, but most of the case we have proline gap, we have some missing fragments, we have different problems. But will it be easier for us and if we have some database which we can compare our spectrum to, right? Even if we're missing something, if it's 95% similar, then probably it is this thing, right? That's why actually for proteomics we need a database. Because in the beginning of proteomics there was an idea that it's only the problem with our mass spectrometry equipment, and we'll get better mass spectrometry equipment and we'll have no need for databases, we just be able to de novo sequence everything that we have here. But not so. And also, if you have just, yep.
May I have a stupid question?
There are no stupid questions.
Yeah, I know, but maybe it is. I'm not. I was believed. No, I believed that these databases are based on data from mass spectrometry.
Yeah. No.
Yeah, I mean sequence of peptides.
Yes, but mostly nowadays we use genomic databases.
Okay.
Even some non-annotated genomic databases. It is not easy to search because we have 3 reading frame on a plus direction, 3 reading frame on a minus direction, right?
Mm-hmm.
But it is still possible to use this information. Of course, I will be talking about some database which is manually created in protein form, so-called SwissProt, which is quite usable, and I do recommend to use it in the first place. And if it doesn't work, then use any other means. But it is usable if you will have some draft of your transcriptome, for instance. Even if you work with some object which is not fully sequenced. I was working with RAI, and RAI wasn't sequenced until the last year. There were only some transcriptomes here, and I was able to work with those transcriptomes without any problem finding all those proteins, proteoforms, and everything I need. So, if this was the question, if I get it right.
So my question was, yes, it is. So in these cases, when we want to specify our sequence, we address to nucleic genomic databases.
And only after you got those proteins, you've get those mass spectra, and you have positive, not just annotation, but identification of different peptides which are present here. You can actually convert it in a protein database and say that, well, nowadays it's manually curated and it can go to the SwissProt.
Okay.
But mostly you, of course, if you are working with mice, human, rice, Arabidopsis, or yeasts, there is no problem. They have already some reference proteomes, and you can get it faster in protein format, and there is no problem. But if you are working with something that is either just sequenced and not quite annotated yet, or not sequenced at all, you will have to use nucleic acid databases. They can be converted in a way to be useful for our proteomics work. So, what we've decided that we can't actually de novo sequence any peptide, and also you have a lot of peptides. It's not always possible to construct the protein of them, because those peptides can be from different proteins, for instance, right? So there's another case why we need database. And also, if you have database, it will allow us to search for those proteins and identify them more easily, with ease, without any problem. So, let us see how will we search our database. Of course, it is possible to search just for spectra, but just imagine a number of spectra you have to make for any organism to just compare those spectra side by side. The size of this database will be enormous, but you can generate those in silico spectra on fly, right? There is no need to pre-generate them. And also you can just compare them, the one that was made in silico and the one that you actually have. As you can see this way, the annotation is quite easy. If you have something similar, you can annotate your peaks and you can see which peaks are true and which peaks are either noise or any other thing. But what will you need? Actually, you will need to have some measure of the similarity of the spectra that was generated from, let's say, genomic database and the spectra that you've got from the real equipment. So, there is problem that we have to now discuss. We've already seen that there are differences in intensity of those fragments, and it's mostly because the ionization efficiency of these fragments is different. Also, what do you think? We have two kinds of ionization which we've used so far. It's electrospray ionization and multi-ionization. What do you think? Which one of them will be more beneficial for fragmentation? And why? What is the difference between those two modes of ionization, electrospray and multi, if we are talking about the ions that we get after this ionization?
In multi, there is single charge, and in EC it is multiple charge. So the EC is more, it can compensate the problems with preference of some amino acids and peptides of ionization.
It can, yeah, but actually it's even easier. If you have some peptide here, which is singly charged, after some fragmentation, only one part of this peptide will have charge and we can detect it, right? If you have a number of charges here, and even if it fragmented in some different way, we can actually have more fragments that we can detect. So for each single peptide, we'll get more fragments that we can detect, right? So electrospray is better in this case, and also for fragmentation, we'll try to use at least doubly charged ions, because this way we'll get more signal, right? Alright, so of course the longer oligopeptides will have higher ionization efficiency because there will be more hydrophobic amino acids there. And actually this amino acid composition is more important than length if we are talking about this efficiency. If you want to dive deeply in this topic, there is a really nice article which summarize all the aspects of peptide ionizations, which will be ionized more or less, and so on. So, database. We've come to the conclusion that we need one. Let's see what database we can use. Again, at the beginning of Proteomics, after the conclusion that it is not possible to just de novo sequence every peptide, there was an idea to make a database of spectra of every possible combination of amino acids, which lead us to this number. If you have only 11 mer peptides, so there's 11 amino acids in this polypeptide, we have this much of combinations. And this combination is excluding any post-translation modifications or non-standard amino acids. As you can see, the size of this database is enormous. It's impossible to use. The other idea was to use information from genomic projects, which actually we do today and use today, and if we'll just take those non-redundant database in which every genomic sequence actually is added after some verification, we'll have about this number of 11 mer peptide, which is more or less possible to work with, although it would be not so easy, it would take a lot of time. But if we'll shorten our list by only peptides which can arise from trypsinization and how we can actually select which peptide can arise from trypsinization and which can't.
Depends on the sequence.
Depends on the sequence, and there are some certain amino acids after which our trypsin make this cut, right? So we know the ending amino acid, at least. We'll have this number, which is already not as big, and if we'll narrow our search down to, let's say, one species, for instance, human species, we'll have just 4 million of possible 11 rare peptides, which is possible to search even on the cheapest laptop nowadays. So there is no problem searching this kind of information. So, what are our conclusions? Our conclusion is that we need a database, of course. This database can be used in different ways. The better this database is annotated, for instance, if it's already in protein format, the better for us, we can use it easier. And if we want to search something, it's better for us to at least know which database is better to search. For instance, we don't need this NR database if we are working with mice, because NR contains everything. We'll better be off with some mouse reference genome, for instance, transcriptome, or for mouse there is no problem, we have reference proteome as well. So, to be able to identify peptide within the database, we need this fragmentation. We need some reliability score to compare how likely that this spectra arise from this protein, which we've found in our database. And also, we would like to use high resolution. We would like to use a low mass mode, so not all mass spectrometers are available for proteomics because of this. And also, we could ignore most of cross-translational modifications because of two things. First, we have some glycosylations. Usually, after fragmentation, those glycosylations will also fragment off our peptide. And if you have really long protein, what do you think? Is there a chance that every serine is phosphorylated here, for instance? No. So we'll be able still to find some information even for highly post-translational modified protein, because there will be peptides which will be the same even if our peptides are naturally post-translationally modified, because not every amino acid which is here is modified. And what's also nice is that if we are working with peptides, we can work with fixed tissue, which was, let's say, fixed in formalin, paraform, whatever, and even embedded in some box for histology, for everything, because even if our proteins now are modified, covalently bind, and so we can use trypsin or other ways to cut it into a smaller fragment, which can be more or less intact, and we can identify proteins from even this archival tissue of some patient, even after, let's say, 30 years of storage at room temperature, which is quite nice, right? How this database search engine will work, and what will we have? First, we need either a protein database or nucleic acid database. The only difference is that if we have nucleic acid database, and if it is not yet annotated, we don't have this start codon which is defined, we don't know open read frame, we'll have to use 6 different read frames, open read frames at the same time. So the database will be more complex. Then, even if you have nucleic acid, we translate it into possible protein sequences. The program can digest it in silico to actually get those peptides, which can be found in reality. We can add some post-translational modifications, and what modifications we actually have to add if we're working, for instance, with gel excised spots. What have we done to our protein in 2D electrophoresis before we get it for analysis?
It is SDS, no?
SDS is washed away, actually, but we've made something to one of amino acids. We discussed it yesterday and the day before yesterday, actually. We've modified this protein by ourselves because we have this cysteines, right?
You mean the
And we've
acetylation?
yeah. We've modified all the cysteines, and we were trying to make sure that we have modified every cysteine here, right? Because in nature it's not always the case, but here in laboratory conditions, to be able to search this database more efficiently, because if we have to search two different mass for cysteines, we'll double the time and resources we need for search, right? So it's not a good idea. We'll try to do so. Also, there is one problem. Methionine is really easy to be oxidized, but this we cannot control. This can happen at random. So we have to actually use this post-translational modification expander and to, after this trypsin digester, change the mass of every cysteine by this added mass, and also make double version of any peptide which contained methionine, either with oxidated form or with normal form. Also, afterward, we can make some filter. For instance, after trypsin digestions, it's possible to get just dimers of amino acids or triplets of amino acids. What do you think? Will they help us to identify a peptide? Alright, let's have that we have some sequence and some characters. We have amino acid which will be cleaved here, right? And will those small fragments be of any, let's some here and here something. For instance, will those two fragments have any meaning to us? Will they help us to search the database?
No.
Will they be in a lot of different proteins, peptides and so on?
Yes.
Do we need to search them?
No.
No. So we want and we can use this peptide mass filter to filter those out. Also, if you know that there is a lot of amino acids which have really low chance of ionization, can we just skip them as improbable to be in our mixture, right? So we can actually compact our probable database just by using this. Of course, it's not a database that's stored on your hard drive. On your hard drive there is only protein FASTA or nucleic acid FASTA, and this is done in memory of your computer by software. Then we can predict fragment ions, just comparing it with the spectrum the actual spectrum which we get from our equipment, and have some score how likely that this given spectra is this given peptide. Are there any questions about how this search engine for protein, for proteomics work actually work? And what are the stages it have to do?
Yes, do we have thus steps in our practice?
Yes, but it will be done stealthily by this mascot server. We'll only need to choose database, choose post-translational modifications, choose which kind of enzyme we use to cut our proteins, and that's all. It will be done automatically by this mascot server. Of course, there's really nice search engines like Tandem X, for instance, Comet, they're freely available. And also there's a really nice search engine called Andromeda. If you ever want to have search engine for proteomics in your laboratory, I highly recommend it. It came as a package which called MaxQuant. This package can have everything you need for proteomics. It can convert your files, it can get your label-free quantification, whatever. And it itself contains this search engine. Unfortunately, because this computer is a little bit, well, you need at least 16 gigabytes of RAM to work with it, so I can't just put it here. But I think nowadays even laptops can have those 16 gigabytes of RAM, so no problem. And this MaxQuant can work either in Linux, which is quite nice, or you actually can use it with Windows because it is written in net.language, so it's possible to actually run it on both platforms. But we, for the easiest, we'll use the easiest possible way. We'll just use this mascot search engine, which is unfortunately proprietary, but it will allow you to make 100 search per day for free. Why not to use it for practical reasons? And also, just a reminder why we want to have those multiple charged precursor ions. Because we'll get more charge on our fragments, and one thing that is not obvious, which I have to mention here, which can be a misleading thing. For instance, let's say that we have some fig here, which we want to fragment. And after fragmentation, we get the following picture. We get some fragments which looks heavier than our fragment. What can be here? Why we have those fragments which looks heavier? First of all, is it Dalton's or M over Z?
M over Z, so
Right.
it is in comparison to,
Is it possible that this is, let's say, 5+ charge? Quite possible. And is it possible that this one have only 1+ charge, and this have also 1+ charge, but it have 4, 3, 4, why not? So, in reality, fragments of your ion can seem bigger just because they have lesser charge, right? And actually, that's what we want in proteomics. We want to use for search only species we have 2 or 3+ charge. It will be more efficient for search. We'll have more fragments. It will be easier to do. Right? Are there any questions concerning databases and how they get searched for proteomics? Let's continue then. As I've already told, there's a lot of different search engines. Unfortunately, I haven't put here Andromeda because I've only been able to work with it last year, and better than most of those search engines. So just small advertisement. MaxQuant and Andromeda works really well.
Why it is so good? So why?
I can't actually. Well, I don't know why.
Because of interface or?
No, the quality of searches are better. So you've got the same data, and it is possible to find more proteins. It is possible to find more fragments of this protein. It is possible to compare them because if you just have the same sample repeated twice or thrice, and you can just put those repetition in your search engine and see what's different because you know that this is the same sample. And with Andromeda, I've got more comparable data than with most of this. So I don't know how they did it. They called it predictive fragmentation something. I haven't actually go deep enough to understand what they've done, but their algorithm is working a little bit better than the others. But all of them are good enough, actually. So there is a lot of different ways, and now we have to discuss how we can address the problem of similarity of our spectra and our search, because actually we can say that everything is well, everything can looks like everything, right? That's just a similarity between them. It's different, and we need such measure to be able to compare between our searches and to say that, well, this is statistically significant, this one is statistically insignificant, or if we just take those three which are statistically insignificant by themselves but put them together, they can get out statistically significant results, right? So I think that you've already know about e-value. You've used it a lot in genomics, I hope. BLAST? Yeah, right. So it's a measure which says you how likely it is to have a false positive. It doesn't give you any information on false negatives or anything, but just how likely it is to get false positive result here. But the problem is that for different search engines, you will have different measures. Absolutely different. You can't compare results that Andromeda provided you with, let's say, Tandem X or this Mascot. Mascot uses a little bit different idea. The idea is that both false positive and true positive have just a little bit different distribution. For false positive, if you'll get here some score, they will be at this side of graph. So actually, some low numbers of similarity will happen a lot, right? And those low numbers of similarities mostly errors, right? And they have this Gaussian distribution, which is no surprise. But if we are talking about correct identifications, they will also have this Gaussian distribution, although it will be moved to more similar scores, and it will be, well, there will be a lower number of true hits than the false one, right? So we have two distributions. Each distribution is a normal distribution, as you can see, or I would say even Gaussian here. And you can provide some value here, which can be used for identification, whether your notation identification is statistically significant and how significant it is. For instance, in Mascot, it is visualized in this way. This part is statistically insignificant, and if you have hit here, you can say that it is statistically significant. It could be that it is false positive, or it could match a lot of proteins at once, because it can be some, for instance, let's say conservative domain, right? But at least this peptide, for instance, matches this spectrum. So let's see what kind of database in proteomics we have. We've discussed database which we can use for our peptide or protein identification, right? And there is, of course, a lot of nucleated databases, whatever you can think of, GenBank, NCBI, whatever. Also, if you have any database which covers your specimen of interest, like SwissProt, for instance, it would be better to use it, because it is already translated. You already have just one case out of 6 cases possible open read frames, which is better. Also, since we've already talked about two-dimensional gel electrophoresis, some 10 years ago there were a lot of different databases with just pictures of those gel and all identified peaks. Unfortunately now most of those bases are defunct because mostly nowadays we use mass spectrometry, and if you want to compare protons, you will just try to identify the differences. You don't know to know all those housekeeping proteins which are at a high concentration, which are mapped on those gels, but it can be beneficial, and some of them, like Swiss 2D page or some, for instance, for maize, also available. Aside from identification by mass spectrometry and gel electrophoresis, there are databases on 3D structure of your protein, because you know that there are some cases of misfolding of proteins, for instance, or that the protein to have some enzymatic function have to be folded in some certain way. Or also, which is quite interesting, some proteins have to change their conformation upon change on pH or any other external factor to have biological function. For instance, if you have some virus which gets into lysosome, will it stay there? If it stays there, it will get proteolyted, right, and disintegrated. It have somehow to get from it. And it happens that most viruses, which actually infect cell after getting in lysosome, have protein that will change its structure, its folding, after changing pH. Lysosome gets this acidic pH, proteins just have some conformation, and virus is released from this lysosome, for instance, right? And that's why just having primary sequence of your protein, even with whole post-translation modification, is not enough for proteomics nowadays. You also need to know about secondary and tertiary structure, and in some cases you even need to know complexes of protein, how they work, how they are organized. And there are such databases. The one that is most known is PDB database, is mostly database which is formed by some Ringel structural analysis, and also there is a database for conservative domains like PFAM, which you probably already used at genomic course to identify mutations, whether they are in some functional domains or in some functional parts of the protein. Also, there are some specialized databases for post-translational modification, like glycylation, phosphorylation, and so on. Of course, we don't just want proteins per se, we want to be able to see which functions of proteins take part in. And I think that you probably already used KEC or MetaPsych or Reactome databases, because those genes and those proteins have so-called gene ontology annotation, which functions and which localization inside the cell they have. And if you have just two proteins, you can have a lot of differences in proteins, and it's not easy to analyze them just one by one. What you can do? You can load this list of differently expressed proteins in the software, which just checks either gene ontology on your proteins or those databases to see that, for instance, the difference that you see is mostly MAPK signaling cascade, for instance, or something that have some relation to cell cycle or whatever. So it's possible to analyze not protein by protein, but just to see what biological functions are affected by this change of protein. And you will have to use those databases. And also, in some cases, you interested in localization of this protein, in which organ, in which states of developmental stages, and so on, those proteins are present. So there is a lot of databases which allows you to see localization of proteins of interest. Actually, this human proteome project I was talking about in the first lecture, they have this proteinatlas.com, which allows you to see a lot of localization of different proteins and different tissues in different stages. So if you are, for instance, working with proteome of, let's say, brain at just some early stages of development, you can search this database, you can see which proteins are overpresent or not present in the tissue you are interested in, and you can plan your experiment accordingly, for instance. Even for some housekeeping genes which could be overexpressed, for instance, at some stage of development. So, a lot of databases, we've just discussed the ones that we need for protein identification, but there are actually a lot of different ways. Let's discuss just some of them. UniProt is the one that is mostly used, and actually this SwissProt database was incorporated in UniProt project. What's really nice that it is a kind of metadatabase. It includes all the links, all the needed information in every of this database. So if you will just open some protein here, you will get links to CAC, to PDB, to whatever database this protein was mentioned in. So it's really easy to have just one point where you can search everything on your protein, even post-translational modification, articles, everything will be there. And what's really nice is that you can convert between ID numbers of those proteins in this database and in other database. There is a special page on the web where you can just put identifier from, let's say, CAC database and get the same identifier for the same protein in this UniProt database, or vice versa. So it is possible to map any of those databases just one-on-one. What we are most interested in is two databases here. SwissProt is manually created, and it is in in amino acid notation, so it's really useful for our proteomics work. And the database which were named 3MBL before, it's, everything that was sequenced goes there, and then only after manual creation it gets in a SwissProt. SwissProt actually have also an archive which includes data from those databases which were previously available. Nowadays it's easier to use this UniProt database, and as I've already said, there is such a relation between different databases. There is European Molecular Biology Laboratory database, which includes almost everything which is sequenced. In America it's CBI, so it's just the same. As you can see, as probably you know, those databases now have crosslinks and everything is included in most of those databases. But this database have both some treasure and some trash. Everything that was sequenced is there. I think that there is no need to search this database if you have some proteomics, unless there are some special cases, right? Which is better that after some, unfortunately it's done algorithmically, it's not some kind of human who does this. All coding sequences are extracted in this TIR-MBL archive. So it's just a presentation which contains only coding sequences, which we can use in our proteomics work, but still there's everything. But it is easier because the we have shorter sequences. Also, after some manual curation and after having some proofs that it's not just coding sequence, but there is some protein or peptide or some other product, it gets to SwissProt database and it is converted in amino acids sequence, which we want to use a lot. And only if there is some conflict in annotation, some error and any other problems, it will get back to 3MBL as a note that there was a conflict or there is a different alleles of this gel, of this gene or anything like this. So the relation between those different databases are more or less understandable, right?
Yes.
All right. We can skip this, and what this database can be used for. We can get this search, and after search we can get this identification. And as you can see, even that I've cheated a bit, I've made in silico representation of this peptide. You can see even in in silico representation, the program decides that some of those fragments will never appear in real data. And we can then, after some search, get this table which includes all the ion species, for instance B and Y series here, and in red ones are ones that were found, because I've asked my in silico generator to produce only this kind of ions, there is only them. And also you can see that there can be other ions which also can add to the information, to annotation or even identification of your peptide. You will get such information after searching in any of the search engines, for instance this one from Mascot. But we need to discuss following questions to be sure that we understand it. Is it enough to have only one peptide identified from your protein?
No.
And if you know that you have only one protein in a gel, for instance,
No.
and if this is a signature peptide, or let's say unique peptide,
We should have at least two.
we should have at least two, right? But if we know that we are working with this animal and in this position in 2D gel should be this protein, and we get this, let's say, annotation of just one this peptide, we can say that, well, probably, yeah, we can at least annotate it, right? So those rules are more or less just common sense rules. But if you have a lot of proteins, if we just work with proteome and we have such identification, be it even this signature peptide, we can say that we identify this protein in any way. Also, what do you think? Is it possible to identify many peptides at the same time using tandem mass spectrometry? The problem is that if we have just one protein and one peptide, it's not a problem. But if we have the whole protein, we just trypsinize it. At the same time, yeah, not probably, but one after another in quick succession, why not? We can just isolate one peptide, then fragment it, another peptide, then fragment it, yet another, then fragment it, right? So the answer is a little bit troublesome here. Also, could some prefractionation of our protein help us in identification of some underrepresented proteins, for instance?
Yes.
If we have, for instance, antibody which can get our protein of interest or post-translation modification of our interest or whatever, we can actually make bigger concentration of our protein interest. Just have a column, have this antibody in this column, pour all our proteome here, and only things that have some affinity to our, let's say, antibody will stay on the column. Then we just change pH or salinity or whatever to get rid of this this antibody to its site of recognition. It will break and we'll have some fraction of highly concentrated proteins of interest, and then we can work with them, right? So prefractionation is something that we can use. Actually, mostly we do this kind of prefractionation by chromatography before entering our sample in mass spectrometry. Mostly, for proteomics, there is chromatography equipment and mass spectrometry which is connected. So, the problem is how to get identification for the whole proteome. The short answer, you could not get identification for every protein in your proteome. The longer answer is that there are two problems at least. One problem is the concentration representation. If you have only, let's say, 100 proteins in your cell of some given protein, you will not able to get enough sensitivity to be able to see them, right? And other problem, we are talking not only about protein but also proteoforms. And let's say that we have digested our proteome, and before that we selected just one protein of interest. So we simplified our task, we have just one protein, but this protein can have different proteoforms, right? And here is a representation of 4 different proteoforms of the same protein. We've got it, we've made this representation, we've get this mass spectrometer identification, but what do you think? Can we say that there are other proteoforms, except for the first one, having this kind of data? No, because those post-translational modifications are missing from our data, right? Due to different problems. And that is a problem that even if you can identify the proteins, it's not always possible to get all the possible post-translational modifications, right? How can you overcome this problem? For instance, you know that the difference is this given protein, but the concentration of this protein is not changing in any meaningful way. It's just the same in your control group, in your experimental group, but somehow by different means you know that this is a protein which have to be the problem.
It has bigger mass, for example.
Yes, you can get the mass of the whole protein, right? And see whether there is difference, because most of those post-translational modifications are big enough, except for phosphorylation, right? Phosphorylation, you can use special dyes, for instance, to make a 2D gel, and you will have those two different proteoforms, right? So there is also a problem, and that's why we have 2 different approaches to see our proteome, so-called top-down and bottom-up approach. For now, just the names. We'll discuss a little bit later what each of them means, but one of them will be able to get you the most possible number of proteins, but proteoforms, this one is mostly omitted. And the other one will have not so much a coverage, and will have the coverage of a 2D gel, no more. And you will be able to get all the proteoforms which are there. And you have to decide which one of them to use, because there is no universal instrument in proteomics, unfortunately. So, if we want to do this top-down experiment, and we want to separate our proteins, or even peptides, we have to discuss a little bit about chromatography. Have you had any course on chromatography or such analytic methods before? Okay, then just briefly, I will just have some ideas. For instance, let's say that we have a bus of tourists. We have some shopping center. And it came that this shopping center is mostly specialized, let's say, in books. We have some time, let's say 15 minutes for our tourists, to get from the shopping mall to different bus. And they have to pass through the shopping mall. What do you think? Will this be separation between those tourists by the chance of affinity to books?
Yes.
Yeah? They have 15 minutes. I hope that they will all get in time here, but there can be, of course, problems. But some of them have no interest in books, or they want to eat, or they want to just get to bus and sit. Some of them have a lot of affinity to books. They want to see which books are there, or buy some books, right? So we can fractionate our bus of tourists by affinity to books. Can we get the same bus and fractionate them by affinity, for instance, for food? Will it be the same fractions? Different fractions, right? So the chromatography basically is the same idea. We fractionate our sample by some affinity to something, or some repellence to something, right? Actually, I want to say that chromatography was invented by Tsvetkov, which worked at St. Petersburg State University. So with proteomics we have a lot of different connections with our city, right?
Цвет.
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 Цвет, sorry, цвет. It was my fault, my bad. Right, цвет. So, actually, he worked in our university. Just chromatography. Цвет, chromo. Yes, we were right, I just... Alzheimer's knew. Yeah, so, the idea is that we get some mixture, and then this mixture gets separated by affinity to some characteristic. There can be a lot of different characteristics we can use, but for proteins, what do you think? Which characteristics we need to use mostly? Weight, length, but for length we have to denotate them, right? So I think the size mostly. It would be size exclusion chromatography or gel filtration chromatography. We'll cover it later. Solubility, right, we can use it, but it's not quite easy. But charge, we can use it for cation or anion exchange chromatography. You were right here. Also, hydrophobicity, right? We can use it as well. These are the main three different ways, and also we can play around different interaction between proteins. Protein and antibody, protein and other protein, and so on. And also have affinity-based chromatography, so-called. Let's see what we can do here. We can either collect our different fractions and then use, for instance, multi-mass spectrometer, because we can just put every fraction on this plate and just get spectra here, or we can connect it to electrospray ionization mass spectrometer, just straight connection. And also we can pre-fractionate our sample by first running some gel-based experiment and then use this chromatography, which allows us to simplify a protein even more. The problem actually with complex proteome is that we'll have either big or small signals, and we can lose this small signal if there is a big signal, or we can have a peptides of the same mass, and if we'll fragment them in the same way, we'll get a number of fragments from those two peptides, and we wouldn't be able to identify them at all. So if we can pre-fractionate them, we can easier identify them. So, which kind of chromatography we are using? Mostly it's reverse phase chromatography with so-called C18 stationary phase. And what we're trying to see is how hydrophobic our protein or peptides are. Also, we use size exclusion chromatography, ion exchange chromatography. Size exclusion is actually the size of this molecule, not only its length, because it can be small in length but have big cross-section. Also, ion exchange chromatography for basic and acidic proteins, we can separate them. And affinity chromatography, we have antibody or just one protein which interacts with some another protein, we can also enrich it. There's other types of chromatography which we are not using extensively in proteomics, like partition chromatography, normal phase chromatography, is mostly for lipids. Displacing chromatography, we will not be covering them. But let's cover reverse phase chromatography. Reverse phase chromatography have really simple stationary phase. It's some bits of silica to which 18 CH2 and CH3 here groups are linked. What do you think? This molecule, is it hydrophilic or hydrophobic? Is there any charge? No, so it's hydrophobic. And if now you will have a mixture of your mostly peptides, protein is a little bit too much of hydrophobicity, it is probable that some protein will stick to this stationary phase and will end up here without being able to dissolve. So mostly C18 is used for peptides here. But if you have some hydrophobic amino acids in your peptide, first of all, peptide is more or less linear, right? It doesn't have a lot of secondary structure. it can form here hydrophobic pocket, right? And since here we have water with just some small addition of, let's say, acetonitrile, this acetonitrile is really some small concentration at 95% of water. It will stick to the stationary phase. Of course, there will be cases of dissociation and reassociation. And if you have a column which is packed with this C18 phase, it will move with the flow, but it will move really slowly. Days and months will be needed to get this protein with high hydrophobicity to come out of column. But what if we'll change this mobile phase, this solution, and let's say have 5% of water and 95% of acetonitrile? What will happen? Acetonitrile can easily dissolve this hydrophobic interactions, and our proteins, peptides are soluble in acetonitrile, and they will be able to get from this column. It's like if you have the same idea of tourists and buses, and you have a tour guide which just gets behind them and tries to get them from those shops, right? You've seen this scene in our city, I think, a lot, and there's some tour guide we have, come here, we have a deadline, we have to go, so that's the kind of thing. And we also can use so-called gradient chromatography when we have this number of water here and acetonitrile here, and this number at the end of our run. Of course, it will not change your resolution or anything, but it will make your peaks sharper. If you have, let's say now, peaks of concentration of your peptides without just this gradient in so-called isotropic, isochromatic regime of your chromatography, you will have some peaks, then some peaks that will be sharpened, and I will have to continue here. But if you will change the concentration in the same time of your, let's say, acetonitrile in this way, this is gradient, what you will have, those peaks will be shorter and they will be higher, because the same amount of peptide will came out of the column in shorter time. So, and also it will move here, right? By using different gradients, different conditions, you can get higher peaks, slower time, and even if you have just say 1 million proteins, it will be enough to get this protein identified in your mass spectrometry, because you will have high concentration is just small time, but you have to have a mass spectrometry which can work really fast in this case, right? So that's about this reversed phase chromatography. Also, we can use this for proteins, but we can't use this kind of modifier in our stationary phase. We'll have to use C4 or C8, because in other case our proteins will prefer to stick here. But those shorter fragments will allow our proteins to get to the column eventually. So, was it clear about reverse phased chromatography? Let's then go to this picture. Actually, there is some theory about the volume of your mobile file phase that you need to use to be able to get good separation of your analytes. Like here, all the volumes given in a column volume. So the volume that your column will have if there will be nothing inside it. For instance, if you have just this acetonitrile here at high concentration and start to apply another round of peptides here, they won't be able to bind to your stationary phase. They will go away. So first of all, we need so-called equilibration of your column, about 10 column volumes of this first concentration, just to make sure that any bit on your column have the same concentration of water as nitrile. Then you can inject it with your samples, and something that can bind to this will just go to the column. For instance, salts. As you remember, we have a problem with salts in mass spectrometry, right? Because they can hamper our analysis. With this chromatography, what we can actually do, we can add our peptides on a column and have some minutes of elution where no peptides will elute at all to go into waste, not in our mass spectrometer contaminating with salts, but just in some waste. And then, when all salts are already gone, we can switch it to mass spectrometry. So, as you can see, we can even dissolve our sample on column for our mass spectrometry equipment. Then we have this gradient, which can take approximately, it's not the right numbers, it can be more or less, 10 to 12 column volumes. And then you have to stick here for 5 more column volumes. Why do you think we need this? We have this big concentration of acetonitrile, and we have to hold it for some time, because column is long, and even when we get here, it doesn't mean that this high concentration of acetonitrile get to the end of the column. It will be diluted with what is in the column. It will be something here, and we will not be able to clean our sample from the column in this way, right? That's why we hold it here, to be sure that now we have the column fully filled with this concentration, high concentration of acetonitrile, and all peptides are washed away, even the ones that are really hydrophobic. And then we can get back to this recalibration concentration, and wait for some additional 10 volumes before we can start another injection. Was it clear? Of course, this is, let's say, ballpark. It's what usually done in just usual condition, can be a little bit different, but mostly it's what you can start with. If you want to do chromatography, you can start with those numbers. Column volume can be either calculated or is usually printed in a document on your column, so you know just as, let's say it's some half a milliliter, for instance, and you just multiply it. So, if you don't mind, we'll end up with chromatography and then we'll have a break. Okay, just 5 more minutes. Another kind of chromatography is so-called ion exchange chromatography. Let's see that we have our stationary phase, which is covered with something different. Not hydrophobic, but something that can have charge. We already have a lot of different molecules which have charge. For instance, amino column, we'll have NH3 or NH2 here, depending on pH, and we'll have a charge, right? And if we have some salt in solution, this charge will be neutralized. For instance, we have here a chloride ion, and what will happen in solution of some salt here. What we can actually do is we can, first of all, have low concentration of salt, so almost every charge group is available. And we put our sample on a column. If our sample have some negative charge here, it will stick, right? And we can get all the sample which is not charged either, or have a net positive charge. It will get from the column right away. Then, actually, most of peptides will have some part of this negative charge, so most of them will stick to the column. And then we'll have the same gradient, but not of acetonitrile, but of some salt. Let's be, not always, but for instance, this chloride will compete with our protein. Since chloride is smaller, it's easier for it to compete and then just repel our protein from here. We have the same gradient to make peaks shorter and taller, and we can dilute it from column, and we'll get different fraction of differently charged proteins. It can be either cation exchange or anion exchange, whatever is the charge here. And that's how it can be done. Just an interesting idea here that we have two orthogonally different ways of separation: hydrophobicity, hydrophilicity, and charge. And we can actually do tandem chromatography to have even better separation, right? We can be fractionated by ion exchange chromatography and have here a lot of salt. Then we can go to reverse phase chromatography, get rid of salt, and also separate them by hydrophobicity and simplify our mixture. Even if you have just 10 different fractions, it's already easier to work with such fractions. Size exclusion chromatography or gel filtration chromatography, it is the same. The idea here is a little bit different from what I've already told you about, and it's a little bit counterintuitive. For instance, we have some bits of gel which have some small channels, and you pour here your proteins of interest in this column. There are some small proteins and some big proteins. What do you think? Which one will elute first? Small ones or big ones?
Depends on the picture. Would be
The picture, yeah, but is it intuitive why they do so?
it looks like the small ones will can go through the small canals of
Right,
these
absolutely,
holes and
absolutely. But from just our point of view, right, if you put something in some, let's say, sand or anything, the big ones are the first to, are the last to go, and the small ones are just easy to migrate. But here we have diffusion, which is really fast at small volumes. And this diffusion forces our small parts to go in all possible pores here. And our big parts just can't get there, they're big enough. And they will fall first. So that's what is actually size exclusion chromatography. No gradients, nothing, it's just a flow of your solution and time. And affinity chromatography, the one that I like the best. It's there are a number of possibilities to covalently bind proteins or other molecules to some stationary phase. Like you have some polysaccharide, there are different polysaccharides. You can activate it with nasty chemicals like cyanogen bromide. Even the name of this chemical is a little bit scary, but it's possible to do so. And afterwards you can add proteins which will covalently bind to this column. For instance, antibodies. And what do you have now? Gel, which have embedded antibodies, and you can pour your samples through this gel. Everything that can be recognized by this antibody will be kept in this column. Everything other will be eluted. And then you can change pH, temperature, whatever. There's a lot of different ways to get rid of this antibody-antigen cooperation. You can break it in different ways. pH is the most used one, but still. And you can elute only the protein of interest that was bound to this antibody. But what is really nice, it doesn't need to be antibody. It can be another protein. For instance, you have protein-protein complex. You can have one protein of complex here, and you can elute all the protein and see which proteins interact with the protein of interest, for instance. Right? And then you can do proteomics on this fraction to see which proteins interact with your protein of interest, at least in vitro experiment. We'll also came to how can we do this in vivo. So, 10 minutes break. If you have any questions, please ask them.
Ты тоже очень.
Что, коллеги, совсем замучил?
А?
Совсем замучил?
Вы это смотрите, глядя на мое выражение лица? Это нет, это не, я не знаю, у меня походу давление, если вы не
Понятно. Я тоже чувствую себя виноватым.
Нет, это домашний человек.
Хорошо,
Хорошо.
Полина.
По сути, я более продвинутая версия. Которая помощь.
Пускай не там говорит. А
Не,
можешь мне позвонить, потому что я только сейчас.
не, проще.
Поставь это на да.
Там полно.
Поставь это
Я скажу, что
я сижу.
они пытаются скрыть.
Домой.
Сейчас я пытаюсь слышно. Видишь, что какие-то они скрыть подкастов.
Ну, он же сказал, что второй
Там вроде есть. Там вроде будет. Там будет стоять на машине. Правда, если есть, то прикол. Красив. Стоп.
просто знаю, что у меня имя такое. Часть означает. Успех.
Помогите, это еще розетка.
Она отодвигается.
Это смежная стена с коворкингом.
Он хотел дошел.
Послушай, журналистский централ. Я просто реально сложно было глаза открыть, Саша, ночью, короче, скринять людей, а они у меня такие, как будто, вот как будто в фотографии, значит, Афины прикрепляются к антителам, так у меня веки верхние к нижним прикрепились, и у меня там не было ничего, а тут так заключили. Нет, это не с антителами нехорошие, но вот. У меня не было там натрия хлорид, это вот везде. Я чувствую, как будто, вот, знаешь, как есть это молния. Вот как будто вот эта молния у людей. И ее просто разрывает. Вот так вот.
Спасибо. Будьте здоровы. Alright then, let's continue. So, what we've already discussed, that there are two different approaches to how to see our whole protein. We can either get all the proteins and then trypsinize them in small peptides and then work with those peptides. And working with these peptides, we of course can separate them by chromatography, so we are not working with this whole millions of peptides. We work with some set of them at the same time. But with this approach, we lose information of post-translation modification for some cases. Of course, for some cases we do get this post-translational modification, but we can't quantify it. And also, quantification here is hampered because we can get different peptides for the same protein for different samples, for instance, which is also a kind of problem. And we can get the same information differently. We can actually start with proteins. We can get only some proteins which we interested in, for instance, by gel, by chromatography, by any means possible. And then we are working just with some set of proteins or even single proteins, in which we can get the whole protein mass spectrometry, as you can see here. We can here see acetylation, phosphorylation, and so on. It's possible to do by the multi-mass spectrometry, for instance, or it's possible to do by other means as well. And after that, we can do our MSMS and data analysis. So the difference is whether we can get all proteins, then trypsin them, and then work on the peptide level, which actually the most usable way. We can get a good coverage, but lose some post-translation modifications and sensitivity. Or we can go to the top-down proteomics, when we first separate our proteins and then get some small set of proteins to be analyzed this way. And also we can analyze the whole proteins before the trypsinization to get information of post-translation modifications. And afterwards we actually can trypsinize them and analyze them as well. So, is the difference between those two approaches clear? Also, there is a problem that top-down proteomics, as you can see, is not something that could be done routinely. Because one thing is just to make trypsinization and put every peptide on a column. It can be done without any labors. It can be done automatically. It's just some sample. But if you are working with top-down proteomics, you have somehow manually separate your proteins, either by chromatography, by gel, by whatever means necessary. And of course, it will take a lot of time. And also the coverage of this top-down proteomics is not as high. It's about or comparable to the coverage you will get with 2D gel analysis. Because you need to get some meaningful number of starting material of your protein of interest before you can do all this kind of analysis. But actually it seems that nowadays some interesting ideas and also some addition to proteomics move us from bottom-up proteomics, which is 95% of all works, to this kind of work, which is more laborious, which requires more time, money, whatever, but allowing us to get quantitative information of post-translational modification with ease. So, just another illustration of the same process. Bottom up, we get our proteins, we just digest them, and we do identification by our peptide. Top-down proteomics. We get first separation of our full proteins, then we get either protein or peptide mass spectrometry. Protein mass spectrometry is nice for post-translational modifications, and then peptide mass spectrometry to just pinpoint the actual place of this modification, and of course the data analysis. But good stuff about this top-down is that fragmentation identifies your open read frame. You know your amino acid here. You can get proteoforms and modifications, and you can measure abundance of different proteoforms in different samples. But as already mentioned, fractionation, which is manual work, and low coverage, the same as 2D page job. So, if you will have any proteomics work, then I could almost guarantee that you will work mostly with bottom-up approach. After 10 years from now, probably there will be some modifications of this top-down method, which allows you to streamline it and do the same with just less time. And of course, this top-down approach allows you to get more information on the proteome. So, now, we have some problems. We have a lot of peptides. We separate them by the means of liquid chromatography. That's nice. But we have some difficulties analyzing them. First of all, we can have a lot of peptides in the same time. And since those peaks of chromatography are not big enough, I mean not intensity but time, we have only some time to analyze it. For instance, let's say that we have some peptides which almost co-elute. They have different mass, but the hydrophobicity is, let's say, almost the same. We need to decide which one of them analyze first, second, and so on, right? And if we have, let's say, just our mass spectrometry without tandem mass spectrometry, for instance, we'll have something that during time we have those masses of peptides. Some of them are higher than some other are higher, and so on. It's time, its intensity, it's M over V. So we have somehow to get some rules. Which of them will try to fragment and at which time? For instance, if we know all information, of course we'll try to fragment and get information on green one here, on black one here, on blue one here, and on red one here. But if you don't have this information beforehand, you only have information of rising some concentration of some precursor ions, right? And if the peak is small enough, you can have an idea that it will be getting higher and higher and postpone this time of analysis and analysis somewhere here or even after it is already gone, right? So we will try to describe so-called data-dependent analysis. When you can get those scanned in just MS without fragmentation, then choose, for instance, the most abundant ion and use it for MS. Of course, it will take some time, and your time chromatography will get here, and then you will again get this MS and choose ion, but you can see that ion that you've chosen in previous series is still big enough, but some other ions are already small enough or missing, right? What do you need to choose? Will you choose the same ion the second time? So you have to exclude this ion for some time, for instance, and get the second highest ion, and also make an MS from it. So you need some rules which allow you to choose which ion to analyze, which of those peptides to analyze at which time. Usually those rules are simple. Just get the most intensive ion and then put it in temporary blacklist for, let's say, 10 seconds. So 10 seconds you will not analyze this mass even if it is the highest one. But you can see that this approach already have a lot of drawbacks. For instance, this time, for instance, our green one is the one that is most abundant, right? And we will try to fragment it here. And then this one starts to grow, and you will try to fragment it here, which is not optimal. Then you will have your blue one. You will fragment it somewhere afterwards. Then in this time, this ion is in temporary blacklist. They will not be fragmented. And you have this ion which happens here, and you will fragment it not on the top, but somewhere here. And since 3 seconds or 10 seconds is not yet ended, you will not fragment anymore. Is this approach feasible to get high sensitivity? No. Also, there could be another ions which are not peptide. Do we not fragment them? For instance, there is something that present in your water or acetonitrile, and it eludes at every time in your column. Right? If it is in solution, it will be seen at any time. Do we need to analyze those ions? Yeah. And also, there is one lucky thing that if we'll try to draw the masses of probable peptides, we know of the amino acids and we know of peptides, we will see that there are some groups of masses where our probable peptides can be, where our peptides can be at all, because we have only some elements like sulfur, carbon, hydrogen, nitrogen, and that's all, if I'm not mistaken. Do you have anything else in our amino acids?
So for cysteine.
Cysteine is something that is
Cysteine.
what I've already mentioned so far.
Oh, I'm sorry.
So, if we'll just sum up all the masses of all the possible variants of chemical structures with those elements, what we'll get that the possibility of finding them at different m over z is not the same. We'll have some groups at which just the sum of elements can provide us with something that is peptide-like. For instance, if our peptide, we have some mass which lies between those peaks. Is it possible that we have some, let's say, iron or anything other than our peptides? It's not a protein, so there is no active centers, right? So there is no need to fragment anything if it will fall between those gases, right? So that's what called data-dependent acquisition. We have a set of rules, and during the acquisition, during the photography run, we decide upon or basing on these rules which peak gets to be fragmented. For instance, those two are not peptides, we will not interest in them, we will not fragment them. There will be possibly something that will elude at every time some contamination in our water, for instance, and so on and so forth. Also, what is quite interesting, if you want to do MS during electrospinionization, what kind of charge we are most interested in? Wilsonly charged ions would be the ones we want to use for fragmentation. So we can add another rule. We can only fragment ions which have isotopic difference of, let's say, 1 or 0.5 or 0.33 Dalton, which is actually 2 plus or 3 plus ions, right? Because our proteins will mostly get multiply charged during electrospray ionization, but small molecules like SDS or whatever will most likely get only singly charged species. That also will allow us to get better results. But even though this idea is really nice and we've already put here a lot of filters which allow us to choose some of those peaks to be fragmented and identified, what do you think? If we'll have the same sample the second time, will we get the same peaks for fragmentation? Will we get the same set of data? No, absolutely, because it's only rules. For instance, you've injected just some nanoliter more of your solution. Your times will move here just 1.8 seconds, for instance, which is negligible. Also, you will have a little bit different composition of water, there will be a little bit different contamination, and so on and so on. And this data-dependent acquisition have one major drawback. It is not repeatable. It will be more or less, there will be like, yes, you will get almost the same set of proteins, but it's almost not the same, and also it's hard to quantify your protein of interest because you fragmented in different positions, right? And the, let's say, intensity or the number of ions will be different, not only because it is different in solution, but also because we've just get some rules and there is nothing deterministic here. So that is what data-dependent acquisition is, and also if we'll ever have to do quantifiable approach for our mass spectrometry, we also have to get into consideration such thing as ion suppression. If there is something that can ionize better, it can rope other analytes from protons, and they will not be able to ionize, and their concentration will be small, right? So, our data-dependent acquisition, I've already described that we have some rules to select just one precursor ion and get this fragmentation. I've already described that since we have only some elements, there is such an idea that we can exclude some areas of our spectra from fragmentation. Also, we have to get only at least 2 plus or 3 plus charge because it will get us with better data. And that the questions that we need to answer before we will be able to start our mass spectrometry run. Number of charges which are we interested in, 1, 2, 3 or so. Static exclusion, which masses are known contaminants we want to get rid of. Dynamic exclusion, for how many seconds we will not try to fragment the same mass. And also this statistical distribution, shall we use it or not? Okay, actually nowadays still we use this data-dependent acquisition a lot, unfortunately, and then let's start with the results with database. For instance, we've got some data from some proteome. Using this mass spectrometry approach of data-dependent acquisition, we've got some number of spectra, and we can upload it onto this mass code search engine. And what we'll get after this is something like the table of protein which were identified in some time. And we can actually try to see which protein are there. As you can see, there is a group of proteins which is identified, and all of these proteins are actually the same protein, GAPDH, it's right, triphosphate dehydrogenase, but from different species, from human, from cat, and from I don't know what. ATCC, so it should be some cell line, I think. What do you think? Which of those three different proteins is the right one? Here is 1, 2, 3, first, second, and third. Here's U is whether this peptide is unique or not, and here is where this peptide was found for which of those three hits. Can you try to make some interpretation? First of all, what we should like, what we should look for, mostly unique or signature peptides, or just peptides which can be at unique. Let's count. We have 1, 2, 3 unique peptides which are for Homo sapiens, and we have 1, 2 peptides for Hephaestus, for cat. So can we decide which one is true?
No.
No. Actually, it's also possible that there is some contamination in our sample. And the funny thing is that your house pets, even if you are working with gloves and with all the needed foods and so on, can be seen on such an experiment. The small amount of dogs or cats' keratin can get to your experiment. So you should see the intensities, and actually the funny thing that in our laboratory there is one person who doesn't have any house pets. I have cat, my friend have dog, and when we were preparing samples, it was obvious from the identification of samples who was the author of this sample, except for the person who have no pets, because the protein that was identified is keratin from palm troglodytes, from some ape. But it's just because the ape has first position in a database and human the second. So it's not something that she actually has some ape in her house. So the funny thing is that the sensitivity is really good. It is for keratin, which is omnipresent. So here we can decide that if we are working with, let's say, for instance, Homo sapiens, like we have some culture of human cells, for instance, then it's not quite probable that there is a cat keratin in there. So you have to use just common sense in this case. But we have to discuss some other cases. For instance, we have different proteins here, and we have also the tree of similarity between them, which is a hitcher protein from either human, from it's a pig, from horse, and human again, but another protein, which is in database is not hitcher protein, but just stress 70 protein, which is actually the same protein but from mitochondria. So let us try to decide which is which and which is the right one. We have a lot of unique peptides here, which are unique even for this database, which includes human, pig, horse, and so on. What do you think? Which is the right one? Also, we have a number of dupes, a number of peptides which were identified during the run. For instance, if you have really high concentration of some peptides, the peak would be really wide, and mass spectrometer will get time to get it not just once, but let's say a lot of times. So let's us try to make some deduction. What it could be? Which one of them, 1, 2, 6, is the most probable one? First of all, shall we look at some common peptides or mostly for unique ones? Again, unique ones. Let's say which one will have the most unique peptides. It's obvious. No, the last. I think that's number 3 here, right? And number 3 here is from pig. Probably, if we work with pig, why not? But we need additional information. We can deduce just by looking here. Because probably we can use some pig trypsin, which also used, and it can be just purely purified before the application, right? For instance. But in this case, I think that it is the most probable candidate, and as you can see, it's something that is common for all of them three. It has a lot of peptides which were found here. Okay. So, as you can see, that even if you got already search results, you have to interpret them, and it's your responsibility to get those results right. The program will only be able to say the similarity, and then the actual identification is what you have to do by yourself. Let's actually think about different situation. For instance, you are working with human cells and you've got some protein from human cells, but there is also a hit from database for the same protein but from some, let's say, penguin. Is it probable that your sample can be contaminated with a penguin protein? Mostly not, right? So even if the hits are about the same, in this case you will have to use just common logic, right? But if it's cat, dog, probably some contamination. Okay, so the other problem, aside from mass spectrometry here, is how to prepare our samples, because usually we want to have just a tiny bit of our sample, and if we'll try to digest it in solution, just put some solution, put some trypsin, we'll have a problem of extracting our peptides from this solution. And the way how we can get more of our sample, just to get better sensitivity, is to use so-called filter-assisted proteolysis. The idea is quite simple, actually. And if you will do proteomics, I suggest that you will use this type of trypsinization, because trypsinization solution nowadays is not quite used. The idea is that you have some filter in a test tube, which can allow to pass only small, let's say, less than 10 kilodaltons molecules. So the proteins and your proteins will, the trypsin and your proteins will not be able to cross this filter, and it will be somewhere here. So the protease can work on your protein efficiently, and only after there are some peptides, they can be eluted to other test tube to be processed later. So it was called filter assist proteolysis, and I actually think that it's better to use it. The protocol is simple. You will still have to buy this filter. It's not quite pricey. Just as Vivacon, for instance, there is a lot of different trademarks. You have to reduce and alkylate your proteins of interest, because you want that in mass spectrometry. You will just add your trypsin here, and then after the trypsinization, you elute your peptides just with formic acid, with just this concentration. And you will have here mostly your peptides and some contaminants, of course. For instance, if you used SDS or anything other to get these proteins from your tissue, you will also have it here. Now you already know how to clean it. I've ended the last lecture with this zip tip idea, when we have pipette tips which are filled with C18 stationary phase. Now you know what this phase is, what it can do, and you can actually get all your sample by this tip, and all your peptide, since it's mostly water with some acid, all your peptide will bind to this C18, and then you can wash it with deionized water, for instance, or with deionized water with some acidification with formic acid. And afterwards, you can dilute your peptides with acetonitrile. which will allow you to get your peptides already in some organic solution, desalted, DSDS, ed, or whatever you want to call it, ready for mass spectrometric analysis. I don't think that I need to read this kind of protocol. The main steps I've just discussed here. So, some technical problems and some things that I would suggest you to use. We've discussed electrospray ionization last time. What will be the most sensitive way to get your analyte? To use electrospray with a lot of volume, a lot of solvent, or just with very small amount of solvent? Small amount of solvent. As you remember that the flow will allow you to form this plume of droplets which will go to your mass spectrometer. We wouldn't try to diffuse, so almost everything that we will got will eventually get analyzed. This idea is actually really nice for proteomics because we have just small amount, tiny amount, some million of cells, for instance, in some cases, for analysis. And it will have also really small flow, like you can see 100 nanoliters, not microliters, nanoliters per minute. It will allow us to get higher sensitivity than having a lot of sample and really high flow rates. This graph is actually allows us to see sensitivity versus flow rate and diameter of your column. So the column for this nanoflow chromatography is actually just a piece of really small thin glass capillary. So it doesn't look like a column actually, it looks like just thin capillary. And it will allow you to get higher sensitivity. That's how they look like. Just really small tubes which have internal diameter of less than 100 micron in some cases, just really small. And what you can also do in some apparatus, it's possible to combine your source of electrospray ionization and column. So this capillary which is needed to form electrospray ionization actually also can serve as a column. In this case, your peaks would be even better. For instance, the peaks you can get from traditional mass spectrometer, where you have this tip which will get your electrospray ionization, and here is a column which is connected by some thread here. And this is a newer addition where your source is actually the column itself. As you can see, you will get lower noise, higher intensities, and the peaks will be separated better, as you can see. So, nowadays, mostly proteomics done is so-called nanoflow regime with such small columns, which requires quite a small amount of your static material, and sensitivity would be even higher than what was possible with previous ways. Also, another way to get more of your proteome is that we've already discussed that if we have really high separation of our compounds, we can analyze them one by one without any problems, right? We can either get really high resolution and be able to get this difference between our compounds, or we can just try to get them apart to be able to see them. And there is a newer mass spectrometry equipment which includes so-called ion mobility. As you can see, after ionization there is another block here, which is called ion mobility. This is technology. Actually, ion mobility technology is really old. It's somewhere middle of previous century. But the idea is quite simple. If you have, let's say, peptides or even proteins, and after ionization, you pass it through, let's say, some stream of gas. The energy is really small. It wouldn't be possible to fragment them by this gas. But what will happen? If your, for instance, peptide
have really small cross-section. It can go to this gas without any problem. It will be fast enough. But if your, let's say, peptide or protein have bigger cross-section, it will take a lot more time to get through this gas. Of course, it could be gas, it could be electric field, it could be just different ideas, but the main idea is that the cross-section of your ion is what actually matters here. And it will allow you to distinguish even two disaccharide, for instance. As you can see, there is a galactose, but it is connected just differently. So, mass spectrometrically, you wouldn't be able to distinguish between them ever. Chromatographically, it's possible, but also a problem. But if you have this ion mobility, as you can see, cross-section of those two molecules will be different, and you will see it as different peak in this ion mobility part. The same goes with our peptides. We also can get different cross-section for peptides of different sequence, for instance. If they have the same amino acids, but the sequence is a little bit different, they can have different drift time in this ion mobility. So what we can do, we can actually get another separation of our analyte before entering mass spectrum. So it will allow us to have better coverage of our and also, what is quite interesting, as you can see, we here can clearly, distinctively see singly, doubly and triply charged ion. This is drift time, so it's ion mobility, and there is m over z, so the mass here. And you can see that we can easily distinguish them. So we don't need to do this prior work. We can just want to isolate only this part of m over z, and we'll, with some guarantee, we'll get only multiply charged ions, right? Fine. Unfortunately, only one manufacturer can provide this kind of equipment because they patented this idea, which is quite simple and was actually done just more than half a century ago. But still, they have a patent. Bruker, now the only provider of such technology with this kind of ion mobility. Other providers have to devise some other kinds of separation. They're also called ion mobility, but it's not with the gas as here, but with either electric field or some other ways, which is not as effective, unfortunately. But I think that others will also catch up and will use ion mobility in proteomics more. I think I will skip all those technological details, except for one thing, which also differs those newer Bruker machines from others. Is that they were able to add here ion trap. It's, as you can see, quadrupole TOF analyzer. We've already discussed it. Quadrupole selected the ion that we want to fragment, and time-of-flight analyzer allows us to get either our fragments and our ammonium ions, which also can be used for getting the sequence. And what is really nice that, as you can understand, that this machine can at the same time get ions and analyze them. There will be some dead time. During analysis, everything that comes from the column will be lost, right? What they've done, it's quite an idea, that all the ions for short time will be accumulated here. And even if your chromatographic, let's say, peak is not big enough, since it is a time when everything is accumulated in this part of mass spectrometer, you will not lose it, right? And after it, it will get to mass spectrometer, will get analyzed by other. The next packet of your ions will be again accumulated in this trap, so there is almost no dead time. While this is accumulating, this one gets analyzed, and so on, so on, so on. So there is a lot of new additions. Last 10 years were 10 years of, let's say, some
newer proteomics methods, new proteomics approaches like those, and I hope that we will be able to use them, because we have only one machine of this kind, unfortunately, but fortunately in St. Petersburg, so it's possible to use it. And just to get to the end of this lecture, we have to have some practical considerations on sample depletion, and also we have to discuss so-called data-independent analysis, which allows us to quantify our samples and the concentrations of proteins. And the problem that we have here, and why we can't see the whole proteome, is that, for instance, we have a human serum, and per 1 liter of human serum, sorry, it's not a milliliter, it's a liter, but still, we have this much molecules of albumin. But for interleukin 6, for instance, we have only this much. You see the difference in just in numbers. So you can get a protocol, a machine, which can at the same time compare those different concentrations. And if we'll look at a human serum proteome here, and we have concentration range here and different plasma proteins here. Red ones are which are easily discovered by the same human proteome project, for instance. Yellow ones are clinically used biomarkers, which used nowadays just routinely. And the black ones is ones which were discovered in some other articles. As you can see, that biomarkers mostly are in this low range part. And it's obvious why. Those biomarkers are not housekeeping proteins, not something that is overrepresented, but they get masked by those proteins. So we wouldn't be able to see them unless we either get rid of those proteins or enrich those proteins by some selective process. Because it is not quite possible to analyze only 44 million molecules per liter. Its concentration, if we get 1 milliliter, it's just this much, which is almost nothing. So, the idea here, we have our proteome which differs by copy number in each cell. We can get this part of proteome where there is some housekeeping proteins or some other highly presented proteins without any problems. If we have a mass spectrometer with higher dynamic range, we can get closer to this part, but there is a corner which have really small amount, like 500 proteins per cell, which is non-approachable by our methods unless we do some concentration, some selective extraction, or some other methods of preparation of our samples. And what we can also do is that there is some limits. If there is some really small concentration, we won't be able to detect this protein. If there is really high concentration, we will not be able to quantify this protein, because there would be a lot of ions, and our mass spectrometer wouldn't be able to discern between them. There would be just some maximum count of ions, over which we can say whether we have any additional ions there. And also we have 2 points as lower limit of quantification and upper limit of quantification. And if we'll see the same graph here, and this all proteins which we have in a sample, we have only this much proteins which we can identify or even annotate, and only this much of proteins which we can quantify, which have enough concentration. So we have a lot of work ahead of us. With new methods, I think that we have at least 50% of unknowns in our proteome, which we can actually solve for. So, was it clear that we can't analyze the whole proteome at this stage of our technical abilities? And we can't actually quantify a lot of proteins right now, but we can overcome this by sample preparation, by concentrating sample, by hunting for some proteins or protoforms of interest, and so on. And just to continue, if we get rid of 10 proteins in our plasma, we'll have this part, and 9% of those 10% will be also omnipresent proteins, and only 1% will be of what we are mostly interested in, and what is actually those biomarkers or something that we can have good publication on. So, sensitivity of mass spectrometers. Actually, we can get really small amounts of samples, like just some hundreds of femtogram or attomoles. There are some records, of course, like Orbitrap can get over 5 orders of magnitude between the lower presented protein and the most presented protein, but of course it's not always possible. And we can get antibody-based enrichment, which can get us up to these numbers of fold encashment, but not always possible. If we don't know what we are looking for, or if we can't provide supplier of antibodies with enough level of antigen, even synthetic one, we'll never be able to get antibodies for this protein. So it's still a problem. Also, if you will be making your sample by yourself, we've already discussed that there are some things that we should avoid, like salts, detergent, something that will not able to evaporate, and some contaminants like keratin. You can see that we can avoid it simple, and there are some different ways how we can overcome those problems. But again, salts, detergents, and some other chemicals not compatible with our mass spectrometry, and we already seen that keratin can get in our samples quite easily. Actually, I've provided with the real data when there was a cat keratin in human samples. Also, what can be done? We've already mentioned that there is some problem of post-translational modifications, and also there is some problem with efficiency of fragmentation and of detection of those fragments, right? But we've discussed only one kind of fragmentation, so-called collision-induced fragmentation, where when we have this quadrupole, there is some gas in it, and our analyte just kinetically gets this energy by collision with this gas. Actually, gas doesn't collide our analyte in a small section. It will get excited. It will get just an additional energy, and then this analyte will fragment by itself. But we can use different additional ways of getting additional energy to our analyte, either with, let's say, light with some laser. We can use so-called electron transfer dissociation. We can add free electron to our sample, and it will also get into a higher energy state, and it will also dissociate. But what is really nice is that this dissociation would be different if we just collide it with something, and if we add some electron, or if we get some electron, or if we use light for this excitation. So those different modes of collision or different other induced dissociation can add us some more information. For instance, collision-induced dissociation of a peptide which have two sides of phosphorylation here. As you can see, two serines here are phosphorylated. Phosphor can easily get all the charge, and as you can see, we have only one here, only one ion. It doesn't mean that there is no fragments, but only this small fragment with phosphorus will get this charge. So collision-induced dissociation could not be used for this peptide, right? We can't identify it by just one mass. But if we'll use different like electron transfer dissociation, for instance. As you can see, the same peptide will dissociate, will fragment differently. And this way we can have all this C and Z, not B and Y, but C and Z, different series of ions, firstly, and it covers actually whole sequence, and we can even assign those two phosphors to those two serines. So, was it clear that collision-induced dissociation, which we have been discussing, is only one of possible ways to collide, to fragment our ions? And different ways of getting more energy to our ions for their dissociation or for their fragmentation can allow us to get more information. Of course, most of the time we will be using just collision-induced dissociation, which is available in most mass spectrometers, but for some cases, like those multiple phosphorylation of peptides or proteins, other ways of fragmenting your peptides can be beneficial. We can, I think, skip this. We've already discussed the resolution, but just a picture. Resolution is important, though it's not quite important, but important for mass spectrometry, because if we have, let's say, peptide of this mass, if we have accuracy of, let's say, 1 ppm, we will be able to get 5 different ways to interpret this peptide. But if we have sub 1 decimal of resolution, we can assign only one possible chemical composition for this peptide. Of course, sequence is a little bit different, but at least we can be sure of composition, chemical composition of this peptide. But as you can see, at low resolution, there is a lot of different combination. But if we have even accuracy of 1 ppm, it's more or less enough, just 5 candidates, and if we can then just fragment our peptide, we can get not annotation but identification. So there is still a discussion in literature, what is most important, resolution or separation? And what do you think? Actually, there is no answer to this question as yet. There are some who say that separation is a way to go, others say that just resolution will solve all our problems. Actually, this is one of topics for Monday. And if you are interested in those different modes of fragmentation or this discussion between resolution and separation, you are welcome to use this as your topic for your presentation. So, the last big part of our lecture is actually so-called data-independent acquisition. What we've done so far, we've have some rules, we've put some rules to select one precursor and then get some fermentation out of it. But what we can do, we can actually divide all our mass over change ratio into small bands and fragment everything that is inside one of these bands, then in another band. And if this can be done quite rapidly, we can cover the whole M over Z with some reasonable window just in 1 second. What are the problems and what are the benefits of such an approach? First of all, the problem is how we identify anything of the meaningful information from here, because we have at the same time fermentation of many ions, right? And the other thing, will this idea of data-independent acquisition be quantitative? If we know which of these peaks is our peptide of interest, will it be more quantitative than this approach? What do you think and why? Every second we get the whole picture. Here we select something, not always the right thing, and just fragment it, which will be more quantitative. We've already discussed that during data-dependent acquisition, there is not deterministic. We can have the same sample, run in the same machine, in the same environment, and get a little bit different data. Using this approach, what would you think? Will it be a little bit more deterministic? If you'll just every second get this picture. We are not depending on any external factor. We don't need to get any of those rules. We just cut our M over Z in some windows and fragment this window, this window, this window, and get information of all the fragmentation in each window, and we can repeat it quite rapidly, once per second for the whole M over Z, for instance. Right? The only problem that we still have is how we can identify which fragments are from which ions, right? And we can do it mathematically. It's not such a big problem. Let's see. Here we have time, M over Z. Here we have all our MS2 mass spectrograms. And what we can see that those three peaks changes during time almost similar, right? If they change similar, can it be that it is the same compound, the fragment of the same compound? Do I need to repeat this picture on a blackboard or whiteboard in this case? Or is it understandable? If there is another substance, it will also have peak, but in time they will be shifted, right? So if we have some peaks which coalesce, which are at the same time, we can assume that probably they came from the same precursor ion. And we actually try to get such an energy of fragmentation that at least some small amount of our precursor ion will not get fragmented. And we also see it somewhere there. It's not depicted here, but still will be present in our sample. So we'll be able mathematically to get all the compounds that we have and all the possible fragments at the same time. Just a mathematical algorithmical problem. Was it clear? So we can do this, it's really nice. Unfortunately, we still mostly use data-dependent acquisitions nowadays, but it is a future. Already, I would say 15 to 20% of application using this approach. And I think that in 10 years' time, we'll mostly be using this approach. The same idea. We have some ions which coalesce, and probably it is the same components and peptides, it's just fragment of this peptide. And those ones are probably some different ions. It's how it looks like. And here in this series we can actually analyze all those ions. What do you think? Will it help us to identify our proteins better? If there is noise, will this noise follow the same rule of bell-curved shape? No. So we can already exclude some peaks which are not presented here. Also, if we have really small concentration of our protein, since every second we have this, will it be possible to register our protein even if concentration is small enough but still in the limit of detection of our mass spectrometry? And during data-dependent acquisition, we can easily skip it, right? So actually, this approach will get you better sensitivity, better quantification. Of course, it's a problem of how you computationally do this, but there is so-called trans-proteomic pipeline, for instance, is freely available. There is such a software as a Skyline, and there is a lot of open softwares which can do this for you without any problems. Of course, there is a steep learning curve on how to use this software, so it's not something that you just put your mass spectrometer data and get results as it is with data-dependent acquisition, but I think it's doable at least. So, the idea is here, and we have to cover now quantitative mass spectrometry, because actually this data independent acquisition is really nice, and it is more quantitative than what we've had with data dependent acquisition. But if you have, for instance, two samples, little bit different samples, and we'll get mass spectrometry information from those samples. What do you think? Can we compare just the count of ions in first sample and in second sample and say that there is a difference between them, this one is bigger, this one is lower? If not, what can be the causes of these differences, which is not actual sample but something else? For data-dependent acquisition, it's obvious. Data-dependent acquisition will ruin our quantitative approach. With data-independent acquisition, what can happen?
Different time.
Probably, but the error will be small because we'll try to model the distribution as a bell curve. Even if we have just only 3 points, we can fit our bell distribution with these 3 points, so it's not a big problem. Though it is a problem, but not a big one. But what about differences protein? Is it possible that there are 2 peptides which coalesce in one sample but not coalesce in other? Will one peptide try to rob the other peptide of those charges? And will the ionization of this peptide be better or worse? So this matrix effect, it's called a matrix effect, can lead to the difference in ionization. And what we'll get here is that we have this change, but we have to prove that it actually difference in our protein. There's a lot of label-free approaches. It's nowadays such a modern tendency to sell mass spectrometry which can do quantitative analysis without any labels. Not quite so, actually. But every vendor tries to sell one. What we can actually try to do to be able to compare two different proteins using mass spectrometry? Labels. We can have either isotopic labels, right? Those proteins which have, let's say, heavy carbons or heavy nitrogens will behave almost the same in our sample. Or we can label them after we already extracted the proteins from our sample, right? And also label them by some stable isotope. Was it clear, or it's already evening and it's a bit troublesome to understand? So, just to sum up, we can't just compare two data from the same machine on different proteomes, even it's the same name, the same solution, everything the same, even the same person doing so, because of some technical limitation, right? Just the bottom line. And to overcome those limitations, we have to have additional protocols which can label our proteins. We can put here fluorescent label as we have done during our 2D gels, right? We wouldn't be able to see it. But we can label our proteins of interest by some stable isotope. So the mass would be different. Or labeled with some post-translational modification which will be cleaved from it, right? We can have some post-translational modification, chemical, artificial, to our peptides or proteins, which can allow us to discern between them, right? And we have different methods which can get us to this idea. First of all, there is so-called SILAC method, stable isotope labeling method. We can get our two different cell cultures. It's possible to do with cell cultures, it's not possible to do with animals, unfortunately. And one of it is with light isotope, the other have a medium of amino acid with heavy isotope. Then we mix it, just 1/1, purify, tropsonize, and get those differences in mass. So the same peptide will elute at the same time, have the same properties, but on a mass spectrometer we have different peaks. For instance, we can label arginine to have difference of 6 Dalton. So they will clearly visible on our mass spectrum, and we can then actually get the differences between those peptides. The ionization for these peptides would be the same, so the difference in the count or in the number of ion would be only because the difference of the concentration is sampled, right? But the problem, we can't use it on the model animals, right? Because we can't feed animals with a kilogram of arginine heavily labeled. It's just too costly. There are other ways how we can do this. For instance, we can add some label after or before. Let's just have another slide, which is a little bit easier to understand. Let's label our peptides after the trypsinization. For instance, we can put something like this in a solution, and as you can see, this group will covalently bind to all the lysines, like we've had in our 2D gels, right? And we can have this molecule with different size of this group in this group. And we can have two different molecules which have the same mass, but the differences of carbons here and here. What is interesting about this molecule? It will self-cleave after ionization and will have this thing gone away. So before ionization, those peptides will have the same mass, right? Because this label, although it's a little bit different, will have the same elemental composition. But after ionization and after fragmentation, this label will cleave at this region and will have two different masses, because this part of the important region will cleave with different mass. What is clear? So we have some label which just fragments differently, and we can select the same mass for precursor ion, and both of them will be then fragmented. But after fragmentation, we have some small mass. You can see the mass is not very big. Compound, which will show how many of these peptides first labeled and second was in our sample. Is it quantifiable? Of course. We have our sample in the same run on the same machine, with the same problems of fragmentation, with the same problems of ionization, so they are quantifiable. Can we use this approach for, let's say, animals or plants? Of course, we don't need to feed them with some heavy isotopes. Actually, there is no heavy isotopes here, so it even can be bought without any problems, because for some reason our companies will not sell us even stable isotopes. They are afraid that it's something nuclear and so on and so on. I don't know why, because we have a lot of stable isotopes around us, but still, this thing can be bought without any problem. Another idea, we can, again, after trypsinization, label our samples with some addition and then somehow fragment it. So all the different ways to have this quantifiable approach is to add some label to our sample that can be visible in our mass spectrometer. But this label shouldn't change mass of our compound unless we already done our chromatography, our first round of mass spectrometry, and only in tandem spectra there should be some differences. So that's how it works. There are iTrack, there is CLAC, there is iCat, just different ways to label them. The idea is actually the same. I don't think that I will have to cover it. If you will ever need to use this approach as a quantifiable proteomics, you will just buy this reagent and there will be some protocol on how to use this. The idea is some label, which we've just discussed already. Also, with the same label, we can get to see the turnover of protein, newly synthesized versus old one. We've already covered in the first lecture, right? And now we know what was this label. We can use some arginine, for instance, which is heavily nitrogen labeled and has this 6 Dalton more than the one that we, carbon, which is 6 Dalton more. And we can destroy those two proteins just by some peptide which contains this arginine, for instance. We can mix those two, or even if it is a culture, we don't not mix because we have old protein and new protein, and we can get this information. So I still have 6 minutes, so just to add some additional notes here. What do you think, if you already know the protein of interest and only want to quantify it, do you need all this modern mass spectrometry equipment, all those protocols, and so on, so on, so on? We can, if we already know what we are searching for, we can use just triple quadruple without any problem, but to quantify our results, we still need this label. What we can do? We can either chemically synthesize the same peptide with some addition of heavy isotope, or we can actually ask our bacteria or other cells to produce peptide or protein which have peptides of interest with cleavage site of trypsin. And we can just add some heavy amino acids on a solution where our bacteria are living, and we can, it would be cheaper than asking for just some artificial synthesis of a peptide, and you will have your own control. You can get this peptide in known concentration, you can add it to the sample just before extraction, so if the real one will be lost in some way, the one which is heavy labeled will be also lost in the same amount, and you can just compare those two peptides, right? This is called so-called targeted proteomics, when you already know what you're searching for, and you can, even with the use of triple quadruple, which is quite easy to use, get the concentration. Of course, you can use Western blot or something, but what if your protein of interest in some attomolar concentration? Western blot is no option, right? Here, the sensitivity of this approach is quite high because you are hunting just one mass and you don't need to scan anything. You just select some precursor here, you fragment it, and you select just one fragment that has this arginine and the one that you are actually hunting for. So, as we've already covered, that quadruples in this regime has really high transferability of our ions. Almost every ion that came to this quadruple will exit if the mass is correct, right? So the sensitivity is really nice. But what I have to say is that if you happen to work with some model organism like human, mice, and so on, you don't even need to devise those proteins yourself. Just go to this database, this web page, select the protein you want to use, select the mass spectrometer you want to use, and it will provide you with peptides which can be used as a marker for this protein. Even with, it will find the higher ionizable marker, it will do everything for you. So it can be done even automatically. Please try this if you want. And that's, I think, all for today. Are there any questions? Okay, then tomorrow we'll have a short part of lecture that I wasn't able to finish today, and we'll also have topic presentation and some hands-on work on mass spectrometry information. So if you have some laptops, just bring with you and try to make search on this mascot search engine. If it will ask you to sign in, mostly it didn't ask you anything, but it will ask just register here because we will be using this extensively. So, thank you very much for your attention.
Thank you very much. это еще не знаю почему, но вообще я знаю, что это не датикс. Но я не могу говорить о тех, но как он это. Все биологи говорили ион, а сейчас